person, they can.
Means as we are a food technologist, we know that the shelf life of a product is the time that it remains the acceptable to eat within the shelf life. The product will remain safe and retain the desired sensory, chemical, physical, and microbiological characteristics. Understanding the shelf life is an essential assuming the safety and quality of the product. Yes, Lokhandi sir, please mute yourself, sir. Yes, that's why Dr. BK Sakhale sir is going to deliver a lecture on applications of novel chemicals in shelf life extension of the food. He's uh, he's working as a professor at University Department of the Chemical Technology, U.D.C.T., and IC director at the Gopinath Rao Munde National Institute of the Rural Development and Research, Dr. Babasaheb Ambedkar Marathwada University, Aurangabad. He is having 21 years of teaching and research experience. He published more than 75 national and international research papers. He published 32 articles related to the food technology background and 5 technical booklets. He has actively participated in TV telecast and radio talks. He has written many books, uh, 3 books and 13 chapters. He has given more than 79 talks. He completed 4 sponsored projects through DST, MOFPI, AIST, and Dr. BAM University Aurangabad, respectively. He has successfully guided 70 UG students. Please hold on a second. Yes, okay, sorry. He has successfully guided 70 UG students and 5 PG students and 3 PhD students respectively. And currently he is guiding 6 PhD students. Professor Dr. BK Sakale's research areas include post-harvest technology, fruits and vegetable processing technology, food safety and quality, FSMS, extrusion technology, new product development, etc. He has traveled extensively in India as visited Israel, Spain, Maldives for academic as well as the research purpose. He has actively participated in more than 10 special training programs and seminars, workshops he arranged. He received Professor J.S. Sindhu Award in 2018 of AFSTI Mysore. He is the president of AFSTI Aurangabad chapter. He is executive council member of the Institution of the Engineers India Aurangabad Local Center. He is a member of Board Innovation Incubation Enterprise, Dr. Babasaheb Ambedkar Marathwada University, Aurangabad. He is also a member of the Malpractice Committee, Dr. BME Aurangabad, from 2018 to 2023. He is also a member of the DRC, RAC, RRC, and Faculty of the Science and Technology, Dr. BAMU Aurangabad. He is also member of the RAC and RRC Faculty of Food Technology, MIT Arts and Design Technology University, Loni Kalbur Campus, Pune. He is a mentor of Bajaj Auto Bharatiya Yuva Shakti Trust, Aurangabad. He is a fellow of the Indian Society of Agriculture Biochemist, Kanpur, India. He received Best Teacher Awards of the Department of the Mass Communication and Journalism, Dr. BME Aurangabad in 2008. He also received IFCON Awards in 2003 for the Best Paper Presentation in the International Food Conference held at Septria, Mysore. He also received Best Paper Presentation Award in National Conference 2008 held at ICAR, Nagpur. He had received Merit Cum Scholarship at BTech MKV Parbhani and AFSTI scholarship at MTech MKV Parbhani. He was chairman of National Level Paper Presentation Competition at MIT Aurangabad. He is working as a reviewer, associate editor, and editorial member of the various journals and magazines. He is related to the food scientist and technologist India, means that is Septria Mysore. He is also member of the Indian Society of the Agriculture Biochemistry, Chemist Kanpur. He is also member of the NI in Hyderabad. He is also member of the Dairy Technology Society India, NDRI, Karnal, Haryana. Now we all are eagerly waiting for the guidance from the extraordinary resource person of the India, uh, Professor Dr. BK Sakhale sir. Now over to you, Professor Dr. BK Sakhale sir.
Thank you, sir, for nice introduction.
Yes.
First of all, I would like to thank you for organizer for giving me this opportunity to address the audience, food technologists, food professionals on the very recent topic, that is FDP, recent technologies for food processing preservation. Now today I will be talking on the application of the novel chemicals for shelf-life extension of the foods. We all are aware that the shelf life is most important because we are working in order to preserve or in order to process the food to have a better shelf life. Therefore, what is actually shelf life? You are all are aware that when the time elapses between from harvesting to the consumption, we can say that from farm to plate, farm to fork, or we can say that from manufacturing to a consumption. So that shelf life, it needs to be extended, prolonged, so that the food will be available for longer time and we can export it. So this is a very nice topic, novel topic, and definitely I am sure that I will try my level best to add something new, information, knowledge to you. And it has been told that you are from the different backgrounds, different specialization. So I will try my level best how to make it a very simpler way so that this technology can be delivered, can be disseminated among you. So I will try my level best to have a look on this application of the novel chemicals. So slide is visible.
Yes, sir. Yes. Slide is visible. Please press the slideshow button, sir.
Okay.
Double click on that. Yes. Thank you, sir. Continue. You can continue.
Challenges we are facing. First is the just Lokhande sir has already mentioned the sustainability. So we are thinking about whether the food or manufacturing population is ever increasing and the food production or agriculture production is not meeting to that particular level. By leaps and bounds we are progressing, but there is one, we can say, worry that whether the food will be available for next generation or not. Means whatever food we are manufacturing or we are producing, whether it will be enough, that means we have to think about the sustainability. This is a global challenge as well as for the challenge for the India also. Because every day 3,000 acre land is decreasing from the cultivation. So the cultivated land is going to decrease. Therefore, definitely it puts on pressure on agriculture how to increase the production. So that we are thinking about the hydroponics, aeroponics, then vertical farming, everything. So therefore, we require the food for consumption, for export, for surplus production will be useful for the processing. Therefore, we need the enough food, then we can process it, we can preserve it. Therefore, sustainability is one of the issue, and we have to find out the solution for that. What is the solution? Solution is that whether we can preserve that food, those are going into waste, because waste foods are post harvest losses, we can call it as technically, that around 24 to 30%. Therefore that can be saved. Therefore we can also is one type of the meaning, we can say that it will be a saving food means generating food. So this first challenge that we have to find out the solution for that, that is enough food, that is a sustainable food production. And second one is the safe food. Whatever the food we are manufacturing, that should have the quality, quality in terms of physical quality, chemical quality, sensory quality, and therefore nutritional quality. And that food should be free from any health hazards. You might have just listened the talk of the Dr. Lokhande sir about food safety. Food safety is playing a very pivotal role in our food pattern. Therefore, that food should be free from any hazards. It should not have any adverse effect on our health. Therefore, the quality with safety, that is the second challenge that we have to find the solution. Third one is the important is the healthy foods. No doubt it should be nutritious, but besides nutrition, the food should provide the health benefits. Therefore, when if you eat a good foods, that will have the good nutrition, that will lead to a good health. And you are well aware that good food is a good health, provide the good health. That will result to provide the health security as well as the nutritional security. That means we are find out the solution for the coming generation, that the food will provide some health benefits. That is called as a nutraceutical, we can say. And fourth one is the good food. So no doubt food is safe, food is nutritious, but it should have the taste. It should have the satiety value. Therefore, the food should be good. So all these four challenges are I categorized as the challenges, and we have to find out the solution. How we can achieve these challenges. When there is a challenge, definitely there is a opportunity. And therefore, we'll focus one by one. India, we are well aware that when we talk about our strength, weaknesses, challenges. So the second largest producer of the fruits and vegetables in the world. So after China, we are the largest producer. But we are not only the producer, but we are also, in some extent, we are also exporting number of the fruits and vegetables across the globe. But the recent production of the fruits and vegetables are 99 million metric tons and 191.77 million metric tons respectively. Because where production is ever increasing. And last year, 2021, the UNO has declared that the year of International Year of Fruits and Vegetables. Considering the COVID-19 pandemics, because fruit and vegetables are having the medicinal benefits, and that's why it is called as a protective food. And that food has played a pivotal role in order to prevent the COVID, and therefore definitely the fruit and vegetable consumption should be increased. It should be made available to all the consumers, all the publics. So that was the motto for celebrating this international fruit and vegetable years. And definitely the consumption, they have made awareness in order to increase the consumption of these fruits and vegetables in our daily diets. And therefore fruits and vegetables are very highly perishables because high moisture content, they are seasonal in nature and having shorted shelf life. This is most important. So in order to increase the production or whatever the production is there, we have to send percent, we have to save it and we have to convert it into a value added products. And therefore, we have to find out such technology that is emerging and sustainable, adaptable, appropriate, that will convert these perishables into a shelf-stable product. Therefore, limited shelf life and coupled with the inadequate processing facilities, because out of the total production, we are only processing 8%. And value addition, when we talk about value addition also very meager. Therefore, we have to increase the processing percentage and as well as you have to add the value. Therefore, the government of the India is focusing on our Prime Minister Narendra Ji Modi has given one slogan, doubling the farmers' income. Farmers' income can be doubled. It is only possible when we'll convert the agriculture into agripreneurship, into food entrepreneurship, then and then it will be possible. Therefore, the limited shelf life that coupled with inadequate processing facilities. So we have to provide the processing facilities, appropriate technology, adaptable technology, and farmer-centric technologies that will they can process their raw material, particularly in the production catchment area. That will we can save the heavy revenue loss in the country. Now, statistics indicates that every year we are losing 80,000 crore rupees due to the lack of this adequate post-harvest technologies or processing facilities. And therefore, we have to save this revenue and we can convert these commodities into a valuable products. I have taken two major fruits that we are the largest manufacturer and the largest producer in the world. That is mango. That is called the king of the fruits, and India is largest producer in the world. We are near about 53% we are contributing to the world, and we are having 600 varieties. Those are having these processing characteristics, and definitely we are also exporting the fresh as well as the processed mango products. Tomato is one of the leading vegetables, fruit vegetables. A major horticulture crop, ranks number one among the vegetables. But due to the lack of the post-harvest technologies and post-harvest management systems, during the supply chain, we are having a very heavy loss in revenue. That revenue for the growers, wholesalers, retailers, exporters, and definitely the surplus amount will be not available for the processing. Therefore, how the losses occur. So therefore, number of the stages we have to think, particularly maturity, ripening, and senescence. These are the three stages in the life cycle of the fruits and vegetables that play a very important role. particularly we require the fresh fruits and vegetable in the ripening condition, which is edible form. Therefore, ripening process of the climatic fruits is highly regulated by the ethylene productions. If you control the ethylene, definitely the shelf life of the perishables can be drastically, significantly, considerably prolonged. The ethylene, a natural plant growth regulator that affects the ripening and senescence. And the commercial strategies are based on the avoiding the exposure to the ethylene. So the ethylene can be generated from a number of the sources. You have to also from the even burning of these woods, burning of this diesel and petrol. So whatever from the smokes, the ethylene will be generated and that will also dangerous or explosive in some cases. Therefore, that it should be used very scientifically and or to minimize the ethylene production and action during supply chain. So the in during supply chain from right from harvesting to consumption, during the supply chain, number of the changes occurs because the fruit and vegetables are the live entity. After detaching from the plants, they are just like us and they continuously respire. We can control the respiration rate. That means we have to regulate this anabolic and catabolic reaction by providing us optimized conditions and whatever the actions occurring during this storage, handling, transportation, logistics, during supply chain, we can regulate. Once it can be regulated, definitely we can make it available. We can reduce these losses after harvest. So the new strategies emerge with the discovery and commercialization of the inhibitors of ethylene perception. From this study, it is observed that the ethylene is the main culprit and definitely control the ethylene in some extent. Definitely, we can save our produce from the spoilage or contamination or deterioration. So these novel chemicals, just I have put the example on one methylcyclopropane to control the ethylene production. There are a number of the ethylene antagonists are available in the market. New are coming, novel chemicals are coming up. So I will also have an idea to give an idea about you the how the novel chemicals are playing a role in the controlling these post-harvest losses. The chemicals interact with the ethylene receptors. There are different mechanism, modes of action of the each chemicals and every chemicals is having the different mode of actions and thereby prevent the ethylene dependent responses. So this ripening is a very genetically monitored phenomenon. Therefore, we have to control at cellular level. So the exogenous and endogenous ethylene can be controlled and we can extend or prolong the storage period or keeping quality. It is therefore need to study the effect of application of novel chemicals on extension of the shelf life of the perishables in general. When we talk about the food losses, every year around the globe, 1.3 billion tons of the foods is lost or wasted. Four of the reasons are there. One of the reasons are lack of facilities, climatic conditions, then skilled manpower. So these can be controlled by using certain strategies, post-harvest strategies. And one third of the all food produce for the human consumption means we are losing. On one side, we are facing the problem of malnutrition. Another second, food industry is waiting for the raw materials in quantity. And third one is the we should have the good quality fresh fruits and vegetable as a protective foods. Therefore, food loss and waste also amount to a major squandering of resources. These are the natural resources we are losing, and that also including the water, where water required for growing it, land to have invested, energy required for these productions, and labor and capital. So all these four parameters, when we look at, definitely whatever the even farmers are not getting a remunerative prices. So the such technologies we have to find out that will be the farmer centric and that will increase the shelf life and that will be food will be available for the longer period in the market. And therefore, needlessly, we are producing the greenhouse gas emissions, contributing to the global warming and the climate change. So all these are the waste is not only a waste, but it also creates some pollution problems. So we have to think about that. And in these post-harvest losses that may occur at different steps during the marketing chain, supply chain, or during the we can say the value chain. First, losses that occurs at harvest, that is during harvesting at injury, mechanical injury, during dropping, the pressure damage, losses at the packing house, due to the packing house, number of the chemicals are used for coating or for waxing, process and wax damages, because they are continually respires, and if you use these wax, then the recommended doses, definitely their will fruit will undergo the anaerobic fermentation and these spoilage occurs. Then losses during the storage, that is chilling injuries, decay, peel disorders, losses during transport, during bruising, deformation, decay, losses at retail, even decay, softening and wilting that observed when these fruits and vegetables are sold on these street markets or roads. Losses at consumer side, decays also, softening and wilting. We are expressing all these losses. We are seeing, we are observing, and we are also noting such type of the observation in our household conditions. When we keep all these fruits and vegetables in a refrigerator, then we will find after week or 4 to 5 days, we again will find those losses even up to 5 to 10% at our home. So we have to control this one. Scenario of this food losses, when we talk about it, will observe that handling of the raw produce, this is most important, because whatever the human resource are involved in this handling, particularly we have to give this, we have to provide the skills them. Therefore, under Skill India, we are trying to give the training to the farmers how to do the scientific handling of these commodities, when it should be harvested, how it should be harvested, how it is to be kept in shade, how it is to be wiped out, washing, cleaning, sorting, through the many stages of middlemen. So during the middleman, the value will increase. Therefore, there is a tremendous scope to increase the, or to add the value during supply chain. Therefore, processing is mostly done in urban areas, because all this manufacturing, processing mostly done in urban areas rather than in the production areas, that leads to the losses in valuable byproducts. Therefore, non-availability of the cold chain infrastructures. Here, all the perishable commodities are handled just like a food grains. There is no special refer containers. There is no supply chain, cold chain facilities in India. Storage of the raw as well as processed product in production catchments. Sir, therefore, government is focusing on where the production is high. That is cluster approach is there. Therefore, that fruit or that vegetable should be converted into a simpler form. That is a minimally processing. Or we can technically, you can talk about the primary and secondary processing can be carried out in the catchment area, in production area. So the cluster development project of the government of state government as well as central government is emphasizing. Therefore, non-uniform agriculture produce due to the fragmented land holdings is not suitable for processing. So now in raw material, when we talk about, we will find out the variation in the each commodity. Therefore, for processing, we require a uniform quantity and quality as well. And therefore, such type of the recommended varieties should be cultivated. That should be recommended from the agriculture universities, and those are involved in this breeding. So such type of varieties, those are recommended for processing, needs to be cultivated. The losses occurs in different types of durables, semi-perishables, and perishables that are costing around 80,000 crores per year. Losses at the packing house. Packing house means after harvesting, we have to clean, we have to wash, we have to reduce the microbial load. Then certain treatments, pre-treatment, we have to do fungicide treatments, as well as for the waxing or hot water treatment for particularly mango. If you want to export it for the European countries and US, vapor heat treatment is given. So along with certain chemicals we have to use. And if it is given in excess quantity or the treatment is not as per these scientific methods, then we will find out such type of the injuries, that is a chemical spray injuries, and the marketability of the fruit can be get decline. Therefore, such type of the precaution we have to take, and such type of the appearance of the fruit is not accepted. It is not adaptable in these markets. Second is the wax damage. If I use the paraffin wax, beeswax, which is recommended, if it is not given a at a particular dose or not as per recommendation, definitely the excess amount or minimum amount if it applied, that will also it leads to the anaerobic fermentation and spoilage fungal growth we will observe. So the principles of the post-harvest management that we have to look at, that is product quality maintenance. Whatever the wholesomeness of the fruits and vegetables, for which purpose we are consuming it, that should be maintained. That quality in terms of physical, chemical, and nutritional quality, and we have to reduce the losses. Means we have to maintain the quality and quantity as well. But to generate the product added value from harvesting each unit operations during supply chain that adds the value in the product. Yes, sorting ingredient we increase the 25% value additions. If you just use the modified packaging boxes, then that will add the value. And also it generate the market opportunities. If you increase the shelf life, then definitely we can tap the distant market, domestic market, and also we can export our products. So these are these facilities that we can have. Then inappropriate post-harvest handling. If a scientific handling is not carried out, then what actually will lose, that we will have the product loss in terms of the quality decay or physical loss. High cost and low profits, because the energy cost is very high for growing the crops. If it is not getting a remunerated prices, definitely will get the very low profit. So therefore, after harvesting, there should be a application of these post-harvest management systems. A loss of the market opportunities, because this shelf life or the life of the produce will be very short. They will have the shortest shelf life, and so we cannot tap the distant markets. Low competitive prices, and if it is not maintained, then we will get the minimum price. The fruits and vegetables as a fresh or perishable products, and we have to take due care during this, because it is a live entity. A number of the physiological processes during these handling and storages. First is the respiration. Is a live entity, therefore it continually respires. We cannot stop the respiration. We can control, we can reduce the rate of respiration. That can be achieved by a number of the ways. Then transpiration, after harvesting due to the continuous respiration, the weight of the fruits or vegetable will get reduced. That we can technically term as a physiological loss in weight. Therefore, we have to control the transpiration. That's why waxing we are recommending, and therefore the respiration rate can be controlled, and transpiration, the losses of the water from that will affect the texture of the fruits and vegetable. Third one, the ethylene production. Ethylene is the main responsible for the converting these mature fruits into a ripening form. And then if it is continued production is going on, ethylene production, it will result into a senescence. Therefore, maturity, ripening, and senescence, these are the three unavoidable stages in the life cycle of the fruits. We are more interested in the ripening stage because it is an edible stage, and therefore we are applying certain technologies, we are using or we are adopting certain emerging technologies that will be a cost effective, having a technologically viable and techno-economically feasible. So the maturity process, though, when we harvest the produce, we have to think about the optimum maturity or commercial maturity. The maturity can be defined on the basis of the end users. In case of vegetables, immature fruits are called as has a gaining importance. So the ethylene is a number of the fruits and vegetables are dependent on ethylene. The climatic fruits in a simple way, we can say that the fruits, those are ripe after harvest, we can call it a climatic fruit. Means their ethylene peak will be observed after the harvesting. So the climatic fruits are sensitive to the ethylene. Use the larger quantity of the ethylene in association with their ripening. That is called autocatalysis. Every mature fruit has to ripe, and every ripe fruit will go to the senescence. So this is the autocatalysis process. We cannot stop it, but we can retard, we can regulate it. In case of non-climatic fruit, most of the vegetables comes under the non-climatic fruits, and citrus fruits are the non-climatic fruits. Those produce the very small quantities of ethylene at high concentration, produce the degraining and increase the metabolism. Particularly in citrus fruits and grapes, berries will find out such type of the non-climatic fruits, and they are not ethylene dependent too much. Leafy vegetables are highly sensitive to the ethylene. Therefore, the yellowing occurs and withering occurs, wilting occurs. And also leaves, particularly coriander, then fenugreeks, will find out when we want to export such type of the vegetables, then there should be no yellowing and withering. Therefore, ethylene playing a very important role. We have to use such type of the ethylene antagonist so that we can keep all these our leafy vegetables fresh as much as possible. The ripening process, it is a physiological process that occurs at the cellular level. So I already explained that it is genetically monitored phenomenon. Finishing the anabolic process, a series of the catalytic reactions starts. Degradation of the chlorophyll, aromas, organelles, and finally causing cellular collapse or death. That is, we can call it as a technically senescence. Therefore, we have to avoid the degradation of this cellulosic material. Therefore, we have to apply the certain chemicals, or we have to create an atmosphere in such a way that will also retard the rate of respiration, so the cellular degradation can be prolonged, can be extended. Therefore, we have to apply this post-harvest technology in order to delay as long as possible the tissue disintegration or senescence phase. And therefore, a number of these technologies are available. Those are novel or the emerging one. So I will focus on these certain technologies. This ethylene is a plant hormone, regulates the wide range of the plant developmental processes and the environment. So this ethylene is very sensitive to the atmosphere. Therefore, oxidation injury caused by the reactive oxygen species is an important factor leading to the quality deterioration by affecting the cell wall disassembly in pulp tissue of the fruits. Therefore, the quality of the pulp is dependent upon the how the gradual change in occurs in the fruits. Definitely, overall development occurs. All these whatever desirable characteristics, particularly sensory characteristics, will observe in the fruits or vegetables. The process of membrane degradation initiated by the action of the phospholipase D PLD enzymes during ripening. This enzyme is a very key enzyme involved in the degradation of this or cellular degradation. Therefore, it leads to the senescence. It also enhanced by the ROS produced during the enzymatic process in stress conditions. Therefore, we have to be very cautious while applying any chemicals in order to prolong the shelf life. When we will see the picture that when a raw fruit is there, raw fruit is sour and acidic in taste, having sugar is in the form of starch, color is green due to chlorophyll content. Pectin, it is hard because of the protopectin, which is a precursor of pectin. It gives hard texture, it is cement-like material, gives hard texture to the fruit and large molecules. But when the ethylene comes in picture, ethylene involves in the various biochemical reactions, particularly the enzymes. Those are kinase, amylase, hydrolase, pectinase, and hydrolase, hydrolytic and pectolytic enzymes that convert the fruit into a edible form. That is during ripening process, there this taste will become a neutral or acidity can be reduced. Then sugar, starch will be converted into simple sugar. Chlorophyll converted into a different types of pigments. If the tomatoes are there, lycopene is there, xanthophyll, then anthocyanins, carotenoids, then less pectin, the protopectin converted into pectin due to the action of the pectin methylesterase, and therefore the fruit will become from hard to soft. And due to the changes in the different enzyme activity, there will be a fragrance or flavor will be there. So all these changes are associated with the action of the ethylene. So ethylene is playing a very important role, which is a naturally occurring plant growth regulator or ripening inducer that affect the growth and development, both the phases, growth as well as the physiological development processes that moves toward the senescence during the ripening and senescence. So can be both beneficial or deleterious depending on the product. So ethylene in some extent it is beneficial for us, and in some extent it is also dangerous one. But in case of the cucumber, particularly when the ethylene spray is taken, then the cucumber, the flowering will be initiated. Ethylene has number of the application, but it depends upon where you are applying it. So in case of cucumber, it initiates the or induces the flowering, female flowering, so that the cucumber yield will increase. But in case of the fruits and vegetables, in case of it leads to the yellowing or withering, and in case of fruit, it will ripe the fruits and then fruit will be moved toward the senescence. The commercial strategies for horticultural products are based on the avoiding exposure to the ethylene and to minimize ethylene production and its action during supply chain. Therefore, in order to make the fruits and vegetables available for longer time, we have to regulate, we have to manipulate the ethylene production and ethylene action during this supply chain or value chain. Then how do we control the ethylene synthesis? There are different ways. At synthetic synthesis level, we can control it. Once it is produced, we can absorb it. There are number of ways are there. Also, we can control the ethylene production by keeping the fruits at low temperature, cooling or cold storages. Then control or modified atmospheric storages. But both these two are having a very costly. Therefore, use of the ethylene antagonist. This is the third one of the ways which is very techno-economically viable. We can use this ethylene antagonist for controlling or regulating the ethylene production. Number of the ethylene antagonists are available in the market. Those few are permitted, few are not permitted in India. On the basis of their toxicity or safety studies, who are comes under the generally recognized as safe. So we'll see the number of the ethylene antagonists are available to delay the ripening, such as the 2,5-narvonadine, then diazocyclopentadine, amino-oxyacetic acid, amino-ethoxyvinyl silver thiosulfates, 1-methylcyclopropane, 1 MCP, hexanol, salicylic acid, silver nanoparticles. These are then ascending order. Those are now the few are the novel one, and that we will see how it, particularly certain two non-boronidine and diazocyclopentadine has ability to control the ripening and softening of the fruits. So texture is most important. And however, none of these compounds are commercially acceptable due to their toxicity and manufacturing concerns. So in few countries it is permitted, and most of the developed countries and developed countries it is not permitted. So the effectiveness of the other compound with potential use, such as the AOA, amino-oxyacetic acid, amino-ethoxyvinyl glycine, EVG, and silver thiosulfates, their use is reduced if the external sources of ethylene are available. So such as climatic fruits, fungi, propane, and cigarette smokes are also that leads to these its effects on these fruit ripening. The ethylene is meant how the ethylene is synthesized. These mechanism of ethylene. So if you control the enzymatic activity at cellular level, so we can control the ethylene production. So the ECC synthase, ECC oxidase enzymes are responsible for the gene expression technology when applied, and definitely the 1-amino-cyclopropane, 1-carboxylic acids, all these are if we control at cellular level, definitely ethylene will not be synthesized or produced. So at a production level, we can control ethylene synthesis. Now the novel approach, we are thinking how to we can prolong the shelf life. There are three different modes of action of the ethylene antagonist. First one, that three will result into a delayed fruit ripening. Number one, blocking the perception of the ethylene, where the ethylene get attached on that receptor site that we can block. So there are a number of the gaseous compounds we can use where this receptor site can be blocked. Then second one is the blocking the expression of genes induced by ethylenes. We are seeing how the ethylene is produced. So the gene responsible for this mechanism, we can control it or block it. Third one is the blocking the ethylene synthesis. So the three different mechanism we can use. So each chemical is having the different mode of actions, but all in total, they will result into a delayed fruit ripening. How the benefits or advantages we can say for improvement in the shelf life and quality of the fruits. If you use these novel chemicals in order to increase the shelf life, what are the benefits? The benefit that significant reduction in weight loss. Weight loss not is simpler way. the physiological loss in weight. So we can say the percent PLW. So the fruit will become will looks like a fresh, maintains the freshness of the fruits after harvest. It improves the marketability of the fruits because it improves the appearance. So the consumers will buy the fruit because on the basis of their appearance. Then improved sensorial quality. The fruit will ripe but uniformly ripe. Therefore, the sensorial quality will be improved. Retention of the nutritional value of the fruits. Whatever nutrients will not be lost during this transpiration or the evapotranspiration because the PLW can control it. Maintaining the textural characteristics. Textural characteristic can be maintained. Then value addition, it will add the value and it will enhance the shelf life and it improves the competitiveness of the fruits and vegetables in the global market. So whatever we are saying that the statistical data, it indicates that out of the total production of fruits and vegetables, only we are exporting only 1.5%. Because whatever the requirements are there from the importing countries, we are not meeting that requirements because our quality of the fruits and vegetables are not that up to the standards. Therefore, that use of this such type of these novel techniques that will also achieve the quality and definitely safety as well. We'll say the first chemical that is hexanol. The hexanol, a naturally occurring volatile cyclic aldehyde, has been observed to be a strong inhibitor of phospholipase D enzymes. And that enzyme is responsible for the key and responsible for degradation of the cell wall. And technologies for its application for enhancing the shelf life and quality of the fruits. We have to increase the shelf life along with maintaining the quality. And therefore, the fruits, vegetables, and flowers, we can apply this chemical for the extension of the shelf life. Are currently under the development. So this is new chemical. So I have also conducted some pilot studies on this chemical on particularly tomatoes or the hybrid tomatoes. This is a mechanism, the hexanol, how it can works, and therefore we are more interested in CTR1 that remains bound to the receptors and signal transduction actively inhibited. So the gene expression will be blocked and inhibition of the ripening. But in case of this, when phospholipid is converted into phosphatidic acid, that will be again gene expression will become active and promotion of the ripening, the ethylene will be released and that will be helps in ripening the foods. Second one, the chemical that is salicylic acids, that is having enormous application in the post-harvest. And the salicylic acid is the endogenous plant growth regulators. It is not new one, it is a plant kingdom. It has been found to generate a wide range of the metabolic physiological responses in plants. Therefore, climateric as well as non-climateric fruit give the response to the salicylic acid. It is considered as a natural and safe phenolic compounds, exhibit a high potential in controlling the post-harvest losses of horticultural crops, and also delay in ripening through the inhibition of ethylene biosynthesis or action. It has two mechanisms. It will inhibit the ethylene biosynthesis and also action of ethylene on the fruit. This is the mechanism from salicylates that has a three application when these low oxidase and enzymes that will be get reduced, inactivated, and then the ethylene production will be reduced. Thus, it will delay the fruit ripening. And after application of the salicylates, that will reduce the respiration rate. Once the respiration rate is decreased, and then the physiological loss of the waste also can be controlled, and therefore weight loss will be decreased, and that means you delay the fruit ripening. Means you can retard the rate of respiration. And third one is the salicylate has the capacity to inhibit the enzyme activity, that is pectin methylesterase and polygalactonase enzyme. This is a peptolytic enzyme responsive for degradation of the cells or the texture, and the fruit becomes softer so that the enzyme activity can be stopped, and definitely it will give the fruit and vegetable and firmer or tender. Thus, it will delay the fruit ripening. So this is a mechanism of this application of this salicylic acid. Then another very recent, we are talking about these silver nanoparticles. Number of the nanotechnology are enormous application in these different type of the domains of these specialization. Also in food preservation or packaging and food processing, these nanotechnology also involves in characterization, fabrication, manipulation of the structures, devices and or materials that have at least one dimension that is approximately 1 to 100 nanometer in length. So these silver nanoparticles, a number of the nano emulsions, nano solutions are available for the post-harvest application as per the requirement of different fruits and vegetables. So the study is going on, and in some certain fruits and vegetables, those are highly perishables. We are trying, and also we have undertaken some limited studies on some mangoes and tomatoes of these locally available varieties. And the silver nanoparticles are the nanoparticles of the silver of between 1 nanometer to 100 nanometer in size and are generally synthesized. I will show the figure how it can taken from the various plant sources, including fruits and vegetables. While frequently described as being a silver, some are composed of the large percentage of the silver oxide due to their large ratio of surface to bulk silver atoms. Numerous shapes of the nanoparticles can be constructed depending on the application at hand. Silver nanoparticles can be synthesized by several methods. So the organic chemist or even in our chemical chemistry laboratory, we have synthesized silver nanoparticle from the anola juice, anola extract, and we have applied in the surface of the tomatoes. So I will also show you this, how the results, wonderful results we receive. So they can be divided into three broad categories: apply wet chemistry, ion implementation, and or biogenic synthesis. This is the mechanism how the silver nanoparticles are synthesized. In laboratory, you can synthesize with the help of the organic chemist, or even the such type of the silver nanoparticles are also available. The umbilical officinalis from avla, the fruit extract is obtained and applied the EgNO3 solution. Then we will see how this under the SEM scanning electron microscope we can see, and by using this Aspergillus oryzae strains, then we can improve the yield of these silver nanoparticles, and we can apply a number of the fruits and vegetables. The third novel chemical, that is 1-methylcyclopropane. It is a very wonderful chemicals, extensively used in the European countries and developed countries. During my visit at Israel, I have worked on this methylcyclopropane in agriculture research organization at Volcano campus, Tel Aviv, and they are using for the all fruits, vegetable, and flowers for the export purpose. And recently, this is a novel non-toxic gaseous compound. Its action in gaseous form, but it is available in powder form. It is called a 1-methylcyclopropane, 1-MCP, as one of the ethylene antagonists. It has been commercially used to extend the storage life of fruits, vegetables, and flowers. It is reported to have the inhibitory effects on ethylene action in various cut flowers, potted plants, and mangoes. So I have taken a lot of trials on Alfonso and Kesher mango varieties, and I have tried to extend the shelf life of the mango by applying this 1-methylcyclopropane, and also 3 to 4 varieties we have also undertaken for its shelf life extension. This is the mechanism we can say here. This is the receptor sites. The 1-MCP has a ability to block these receptor sites. So these molecules, ethylene molecule, when they will attach to the site, they will block this compound. Therefore, the ethylene molecule will not have the sites to bind and to induce the signals for the ethylene production. So why the 1-MCP is used? Because global estimate of losses of fruits after harvest are the 30 to 40% in the region of tropical fruit production, mostly due to the rapid ripening. So it can control the ripening very significantly, and so we can save the losses. And this 1-MCP controls the ethylene production. It is a non-toxic gas, interacts with the ethylene receptors and thereby prevent ethylene dependent responses. So the signal will not be sent to the other genes. Therefore, definitely there will be no ethylene production occurs. So easily released as a gas when the powder is dissolved in water. So it is available in powder form. The person who developed this 1-methylcyclopropane has got the Nobel laureate. He converted the gas into a powder form. The tablets are available Its cost is for 1 gram around 3,000 to 5,000. It is required in very minute quantity. You can say in 1 ppm, 2 ppm, up to 1000 ppm. It can be used depending upon the type of fruits and vegetables. And so the optimization of dose is most important. If the dose is very high, there is every possibility that the fruit will not ripe. Therefore, standardization or optimization of this dose as well as the exposure, temperature and time, that also matters. So these are the variables. Therefore, when we use this powder in a lukewarm water, in a closed chamber, then it will release the gas. That gas, that is 1-methylcyclopropane. That gas will come in contact with these fruits to the respiration. Then it will have the 10 times faster capacity than the ethylene to bind that sites. And definitely this is a wonderful chemicals, and it is approved by the it is inhibitor of the ethylene perception, approved by the United States Environmental Protection Agency. It is safe. It is under GRS, and it is marketed as ethyl block, agrofresh or smart fresh. And for a number of the export trials, we have taken these trials, and in Israel, they are extensively using these chemicals. It is commercially used to extend the storage life of fruits, vegetables, and flowers with negligible residues and active at a very low concentration. It interacts with the ethylene receptor and thus prevent ethylene dependent responses. So this will bind these sites of ethylene here. It fits into these sites, and therefore the ethylene will not have a site for binding. It blocks the binding. Therefore, results in dealing the physicochemical changes. Whatever the ethylene associated changes, that will be stopped, that will be controlled relating to the ripening as well as the reduction in decay and weight loss. Extending the storage life of a range of both climatic and non-climatic foods. It has the ability to control the exogenous and endogenous ethylene in both the climatic and non-climatic foods. This is the mechanism of the ethylene synthesis, because the SSG synthase that will inhibit the ethylene synthesis, AO and AVG we have seen, and this result into promote the ethylene synthesis, fruit ripening, flower senescence, indole acetic acid production, wounding, chilling injury, drought stress and flooding. So the SSG synthase if you control, then SSA oxidase if controlled, then ethylene will not be released. Therefore, that will responsible for promote the ethylene synthesis, ripening. So by considering this mechanism, SSG synthase and SSA oxidase enzyme can be controlled. So this is the mechanism of action of this 1-methylcyclopropane. This is the ethylene receptors are embedded in the cells. This is the ethylene receptor sites. This is the ethylene molecule is there. Molecules in the air bind onto the receptor is fits here, and therefore once it is fitted or it blocks the site, it sends the signal. And once the signal is sent, then ethylene gas will be released. Ethylene molecule acts like a key, unlocking the receptors. Once it is unlocked, so this 1-MCV has a capacity to unlock this one, and therefore the ethylene will not be released. So the chemical signal is sent to the cell, and the ethylene molecule releases. And once the ethylene molecule is released, then most of the pectolytic, hydrolytic enzymes activities are initiated. That will leads to the ripening process. So the what actually the mechanism 1-MCP is doing, the molecule of the 1-MCP also bind to the ethylene receptors. So it is in bound forms. However, 1-MCP does not unlock the ethylene receptor, so no message is sent. So it is blocked by these are the ethylene molecules. They are searching the sites, because all the site is, because it's faster, 10 times faster than the ethylene. You apply both the chemicals at a time, then 1-MC has the 10 times faster rate to bind these sites. Therefore, this 1-MCP is not released. So the molecules of ethylene are unable to bind to the receptor. This is locked one. And therefore, the ripening is delayed. This is the mechanism studied, and therefore, number of this research work is going on for particular type of the fruits. We have to standardize, we have to define the application dose, exposure time, and temperature of application. So this is a very novel chemicals, and the fruit and vegetable which are responsive to 1CP. So we can all the fruits, we can use this 1-methylcyclopropane, and definitely we can extend the shelf life. Banana, particularly the perishable fruits, apples, blueberries, Chinese jujube, then guava fruits, kiwi fruit, lychee, mangoes. So I have taken the trials on mango, the Kesar and Alfonso varieties. We have received got the very fantastic results. A number of the papers were published on this one. Then melons, particularly melons, then papaya, peach, pears, all the climatic and non-climatic fruits, they are the response to this one. Then vegetables, broccoli, carrot, then lettuce, potato, tomato we also studied. In case of non-climatic fruit, the grapes, we can apply this, use these chemicals. And third, another technique that is acidic electrolyzed water we can use for the increasing shelf life. So acidic electrolyzed water was initially developed in Japan and has also been described as a functional water. It is produced by the passing a diluted salt solution, particularly sodium chloride and potassium chloride, most frequently used. Though an electrolytic cell within which the anode and cathode are separated by a membrane. Outside of this electrolytic cell from which EW, that is the acidic electrolyte water, is obtained and produces the various chlorine compound that has the inhibitory actions or it can control these spoilage. So making the electrolyzed water, which is the salt water solution, the acidic and the alkaline solutions are stored in separate tanks where they can be used as a cleaner or sanitizers. So cleaning and sanitizer, acidic and alkaline conditions, so we can reduce the microbial load and then we can increase the shelf life. Next one is the ozone, how this ozone O3 will help to increase the shelf life or can keep the quality for longer period due to the possible formation of the carcinogenic chlorinated compounds in water by chlorination. Ozone is attracting more interest as an alternative sanitizer to chlorine. In 1997, ozone was granted generally recognized as safe under the food additives, grass chemicals or grass additives, we can say. Yes, it's safe and non-toxic, has since been fully approved by the US FDA as a direct contact food sanitizing agents and forms of action on the food. So that will also help to maintain the quality as well as it will reduce the microbial load. So how it works, the bacterial cell, which is a can cell membrane, cell wall, water and DNA molecules. So all the during this, when you add this ozone, then lysis of the cell occurs and definitely that will be, bacterial cell will be killed and then definitely the shelf life will be increased. How, but the how now novel chemicals, how it can be applied? It is the most interesting part because selection, we have to select the raw material because which is very sensitive or ethylene dependent. So the climatic or non-climatic fruits or vegetables we can select and at proper stage of maturity because immature fruit will never ripe. So the maturity stage, it depends upon the type of the fruits or vegetables. So vegetable, most of these physiological or horticultural maturity is most important instead of physiological maturity because the tender or the firmer vegetables are always preferred. But in case of fruits, the maturity, the physiological maturity is most important. And such type of the fruits and vegetables are can be selected. Then we can use the number of the instruments for the weighing balance, your refractometer, texture analyzer for a measurement of the textural characteristics, gas chromatography for the ethylene or for measurement, and mineral colorimeter for measurement of the surface. These are the we have to consider while conducting the experiments or when applying. So I have taken few trials. This is the hexanol concentration and 1-methylcyclopropane concentrations, acetic acid concentration that will be optimized by conducting the experimental trials. Because if the dose is very high, definitely there is a possibility of non-ripening of the fruits. So we should be very cautious while applying such type of these novel chemicals. Then exposure time at 8 hours, 12 hours or 24 hours in a particularly closed chamber. It is most important in case of 1 MCP. And the storage temperature also playing an important role, whether it is ambient temperature or low temperature or refrigerant temperature. And dependent variables that include the physiological loss in weight. The weight of the fruit can be reduced so that we can maintain the wholesomeness of the fruit. We can retain the color, we can retain the nutrients. Then texture, Firmness in terms of the texture, the physical characteristics. The fruit and vegetables are known by their textural characteristics. Therefore, the firmness should be maintained. Then external surface color, because during ripening, the first change we will observe that the color of the fruits will be a prominent one or dominating one, from green to the yellow or red or pinkish, or depending upon the type of the phenolic compounds. Then sugar content, total soluble solid, it is one of the sign. All the changes are associated with these ethylene, and that ethylene is also controlled by these certain chemicals. Total soluble sugars, then total sugars, titratable acidity, ascorbic acid content, analysis of the bioactive compounds, particularly the lycopene, or those are having the nutraceutical effects of phytochemicals. We also can study this type of the bioactive compound in their activity in terms of antioxidant activity. Sensorial quality parameters, overall beside nutrition and health benefits, we have to also think about whether the food is having a good sensorial characteristics or not. Then shelf life period, most important, and is we are trying all these chemicals or applying this chemical for the extension of the shelf life. The methodology that we can apply here, collection and selection of the different components used for the making enhanced freshness formulation. The fruit should be fresh or vegetable should be fresh. Therefore, preparation of the different EFF combinations in various concentrations. So we have trial and error number of the trials we have undertaken, and we have optimized this dose, we have optimized the exposure time, we have optimized these storage temperatures, and also in combination with the other modes of this storage, and we have come to the conclusion how the prolonged shelf life can be prolonged. The climateric fruits and vegetables harvested at the various stages of maturity is treated with a specific level of novel chemicals. Because stage of maturity in case of tomato, there are 4 stages of maturity: green, then light red, breaker stage, and then red stage. Therefore, during this maturity stages, ethylene already synthesis has been observed. Therefore, the selection of the particular chemical and selection of a particular fruit, it is very most important, because it should be compatible to each other. Once the ethylene production has been initiated, then we have to see that which chemical is having this exogenous application that will have the controlling or regulating the exogenous ethylene production. So hexanal 1-MCP silver nanoparticles and salicylic acid we have tried for the subsequent delaying the ripening process at a specific storage conditions. And this evaluation of these post-harvest treatments of novel chemicals, hexanal 1-MCP silver nanoparticle and salicylic acid in various concentrations and duration on a climateric fruits and vegetables, particularly appearance, physicochemical qualities, standard quality assessment of climateric fruits and vegetables. Quantification of polyphenolics, we also analyze the fruits, treated fruits for these total polyphenolics. Then anthocyanin content, phenolic compounds by using the GSPLC and GCMS. After quantification of the different bioactive compounds, storage studies of novel chemicals were undertaken and of these treated fruits at controlled and modified storage conditions. And the effect of the 1-MCP treatments on various physicochemical characteristics like percent weight loss, physiological loss in weight, firmness, surface color, total soluble sugar, percent acidity, total sugars, ascorbic acid content was analyzed. The further investigation was seen on the effect of the novel chemicals treatment on the organoleptic characteristics such as appearance, color, texture. This is subjective evaluation of the fruit sourness of flavor, because most of the chemical apply, there is possibility of getting the off flavor. Overall acceptability or consumer acceptance carried out with the help of the semi-trained panel of judges for the consumer acceptance. Now, whatever the data generated during the experimentation, statistically we analyze for their significance. The knowledge obtained during this optimization of the novel chemicals, application parameter, novel chemicals application, the parameters can be effectively used to scale up for the industrial scales. So we also tried our level best to give these trials to these farm to fork solutions enterprise. These applications of the novel chemicals, how it can be applied, we have to select the freshly harvested climatic fruits or vegetables at proper stages of maturity depending upon the type. It is washed, clean, and it is subjected to pre-treatments. Pre-treatment, we have given the fungicide 1000 ppm, Bivestin and Benomil by dipping in 10 minutes. Then we applied the novel chemicals at different concentrations, varying concentration, varying exposure time, and specific storage temperatures. And then evaluated of the physicochemical and sensorial quality characteristics of treated fruits. And we have assessed the shelf life extended by the novel chemicals, particularly these four chemicals: hexanol, 1-MCP, silver nanoparticles, and salicylic acid application as compared to the controls. So we have conducted the experiments. These experiments are conducted during my visit at Israel. In particularly in cherry tomato, we have taken the trials, that is a mature green and pink variety 1402. And this mature green, the first stage is a pink stage of breaker stage. And this is a control treatment. We have applied the 1-methylcyclopropane 600 ppb. Exposure time was 24 hours. And in a closed chambers, this were stored at 12 degrees centigrade and 20 degrees centigrade treatment. So these are the mature green stage, that is a GT1, and GT2 means stored at 20 degrees centigrade, and same with pink PT1, PT2, and these are the treated with MCP, GT3, GT4, and PT3, PT4, that is pink tomatoes. So already where the ripening has been initiated, so we collected such type of samples which are now gradually ripening was observed, and they were stored for 2 weeks. And we have determined these various parameters, that is weight loss, and surface color by using the Minota colorimeter, and firmness by analog durometer. In our laboratory, we have used this texture analyzer. The chemical characteristic, particularly total soluble solids, by using the refractometer, Atagomac, and sensory evaluation by 5 untrained panelists. The attributes were sweetness, sourness, off flavor, texture, and its crispiness. So these number were given for the 1, 3, and 5. And on the basis of the sensory evaluations, the results indicate that the physicochemical characteristics, this is mature green and pink, this is the before treatment, we analyze the fruits, and then after treatment, we explain how the changes occurs of the 1-MCP treatment. This is the, you can see in green tomatoes, 1 and 2 at the control treatment, when the green and stored at 12 degrees centigrade and 20 degrees centigrade, then you will find that when the GT3 and GT4 and PT3 and PT4, these are the 1-MCP treated treatment. In days, we will find that green tomatoes they are having the greenish in color with the retention of the texture. Means 14 days with a very minute quantity, that is 600 ppb concentration for 24-hour exposure and stored at 12 degrees centigrade, 20 degrees centigrade, having the shelf life of 14 days. In case of the pink varieties U1, they are also having the better shelf life. And U1, another 10 to 14 days, they will be available for the market. For sensory evaluation, after this, in a dark room, we have studied under light how the changes in internal sides of the ripening. It is also observed that the changes in appearance of the tomatoes after the 14 days of storage under the red light will find that in case of the MCP treated fruits, they exhibited the internal quality of the fruits is very intact, firm, and texture found a very excellent. These are the weight loss, physiological loss in weight. So the loss in weight observed very gradually or decrease gradually, so that they have retained this moisture in the fruit, so that their textural parameter was excellent. In case of physiological loss in weight in GT3 and GT4, it was maintained at lower, means evaporative losses or the weight loss was found negligible. The tomatoes in green stage could be stored longer time than pink ones, because in pink varieties already the ethylene was produced, but due to the application of the 1CP, the further production has been stopped and the gradual or slow ripening was continued. Weight loss was recorded higher in pink stage than the green ones in both treated and non-treated fruits as well. In case of this effect of post-harvest application, changes in surface color, we already seen the picture how they will have the effect. Control fruits ripen very fast at both the temperature 12 and 20 degrees centigrade as compared to those treated with the 1 MCP. A tomato treated with 1 MCP and stored at 12 degrees centigrade were found to be most effective. If you combine the treatment with MCP as well as storage temperature, definitely will find the good results in delaying the ripening process and maintain the green color at the end of the storage period. In both the stages, that is green and pink stage, 1 NCP with a low temperature was identified as the most effective treatment to reduce the reduction undergoing the value of UV angle, means color. This is again, I will go very fast we will cover within 15 minutes. Then effect of post-harvest application on the changes of the firmness, means after 2 weeks, the fruits were the firmness. The firmness value of the fruit harvested at both the maturity stages, that is green and pink stage, treated with the 1 MCP decline more slowly than the control treatments and remain tightly clustered throughout the storage period. I mean, the degradation of this cell wall was not observed, means these hydrolytic enzymes were inactivated or these gradual change were noted. The control fruits ripen very fast at both the temperatures as compared to the treated with the MCP. So in both the stages, 1 MCP with low temperature was identified most of this effective treatment for the color retention, color improvement. The firmness value for the fruits harvested at both the maturity stages and treated with 1 MCP decline more slowly than the control one. Therefore, 1 MCP treatment was found effective in suppressing the ripening process in green and pink tomatoes. Once green tomatoes, ethylene was not produced, but in pink, ripening process was initiated. But this 1 MCP has a capacity to even control ethylene production or ethylene synthesis as well. At both the storage temperature without any marked increase in TSS content of pink tomato during the entire storage period. And therefore, as far as the sensory evaluation are concerned, this 1 MCP treated fruits found with better overall acceptance with respect to sweetness, sourness, and texture in the fruit harvested at green and pink stage. Advantages of this delayed fruit ripening by the application of the 1 MCP, that we can assure us about the top quality fruits because we are consuming it for the lycopene content. Lycopene should be fully developed or synthesized, allowing the fruit to exude the full quality. Consumers will get the value for their money. Widening of the market opportunity, we can tap the local as well as distant markets and reduce these post-harvest losses during the seasonal glut or market glut. This is the benefits of this, and this is really a breakthrough. These are a tangible output in case of the reducing the post-harvest losses. And for the experiment, I would like to conclude that 1 MCP treatment at 600 ppb for 24 hours and stored at 12 and 20 degrees centigrade temperature significantly affected the post-harvest quality and storage life in both the mature and pink tomatoes. Then comparative studies when with other novel chemicals we have undertaken, and we will find that these are the different four chemicals: hexanol, 1-methylcyclopropane, salicylic acid, and silver nanoparticles. So treatment T4 hexanol 1200 ppm concentration and stored at 20 degrees centigrade storage temperature at the shade, we can say. Then 1 MCP at 2.5 microliter per liter. Then salicylic acid at 200 ppm or microliter per liter and stored at 20 degrees centigrade. Storage temperature was constant and concentration was varied. And we have taken the trial on the tomatoes, particularly the commercial varieties, hybrid varieties, particularly Vaishali, Avino varieties. And we analyze all these fruits for the various physicochemical and sensory quality parameters. These are these parameters we have studied. There's the percent physiological loss in weight, comparative studies we have taken. Then surface color, color development, and fruit firmness, total soluble solids development, titratable acidity, reduction in the titratable acidity, formic acid content, total phenolic content, and lycopene content we have analyzed. Results that we have obtained at effect of various concentration of the hexanol, 1 MCP, salicylic acid, and silver nanoparticles at different concentration with different storage temperature. Effects were studied, and whatever the results were, major findings were obtained are presented for your clear understanding. The standardization of concentration, we have optimized number of the trials, and I have explained you that T4 treatments we have finalized by trial and error, and we have given this concentration to the selected vegetable site by using this statistical design CRD. On the basis of the trial studies, the application of hexanol, both the varieties of tomatoes, Abhinav and Vaishali, the effective concentration 300, 600, 900, 1200 microliter per liter, and then 1 MCP, same treatment up to T4 treatments, and this concentration of all 4 chemicals were found effective when tomato fruits of both the varieties exposed to them for 24 hours and then subsequent storage of tomatoes at 20 degrees centigrade. The treatment temperature we can keep the same, but then we can store at a regular storage temperature. Thereafter, the effect of post-harvest application of hexanol and other 3 chemicals at this standardized concentration of tomato fruits was studied for 4 different quality. These are these effect you can see. The ABC, this is the control fruits, and DEF, these are the hexanol treated fruits. So the shelf life of the EF, that is you can see the 28 days we have extended or prolonged. In case of the 1 MCP treated fruits, the shelf life of we have extended the shelf life up to 34 days, 20 degrees centigrade by application of the 1 methylcyclopropane at 250, 2.5 ppm, that is 2500 ppb concentrations. Storage temperature, exposure temperature are 24 hours, and storage temperature are 20 degrees centigrade. The salicylic acids, again you can see the effect is the control treatments, and these are the treatments with the salicylic acids, and we have extended the shelf life up to 32 days respectively. In the silver nanoparticles treated fruits of tomato fruits, you can find that the wholesomeness were also maintained, and the shelf life up to 28 days we have observed, and these are the fruits having the freshness or wholesomeness, or we also retain the quality characteristics as well as the nutritional characteristics. This effect on firmness of these tomato fruits, that is in terms of the texture, by using this texture analyzer, which was measured as gram force, possesses major variation among all the treatments of control and treated samples. Moreover, 2.5 microliter per liter 1 MCP concentration, the sample remained fit for consumption till 34th day and also retain maximum firmness, which is both the cultivar Abhinav and cultivar Vaishali. These are the hybrid varieties, and earlier in Israel I have seen the cherry tomatoes. Those the thickness of the skin is different, peel thickness is different. Therefore, dose can be varied as compared to the rest of the treatments as well as the control samples. You can see how these storage period and firmness graph, you can see how the effect of this 1 MCP treatment on the firmness, effect of salicylic acid on the firmness or retention of the texture, maintain up to 32 days of storage. Silver nanoparticles also up to 10 to 8 days we have maintained the texture of the fruits along with the colors. From this, that we can conclude that from this investigation that we have conducted, that 1 MCP at the concentration of 2.5 microliter per liter found a superior as compared to the all treatments of the rest of the 3 chemicals. Among these 4, these novel chemicals, the 1 MCP is found very effective in both the varieties fruits. Shelf life of up to 34 days. The treatment also found most efficient with respect to the maximum retention of the overall quality characteristics, including all the physicochemical parameters. In toto, this treatment was found effective. Then the tomato fruit treated with 200 microliter per liter concentration of the salicylic acid recorded the shelf life 32 days. This is the second chemicals found effective, which was higher as compared to the remaining treatments of hexanol and silver nanoparticles. Therefore, the maximum shelf life recorded by tomato treated with the most effective concentration, 1200 microliter per liter of hexanol, was 28 days. For silver nanoparticles are effective at 28 days. Therefore, we can conclude that the control samples of all the treatment showed the shelf life of maximum 12 days. Means against 12 days, we can extend to the double or 2.5 times we have extended shelf life at a very nominal cost. So the list among all these treatments for both the varieties of tomato fruits. It can be concluded from the present study that 1 MCP treatment concentration at 2.5 microliter per liter and 20 degrees centigrade storage temperature maintain the shelf life of tomatoes with better retention of the overall physicochemical parameters and functional characteristics, such as the total phenolic content and lycopene content as compared to the other treatments. The post-harvest application of 1 MCP was also found techno-economically feasible as compared to the treatment of the rest of the chemicals. So overall, this summary, I can conclude that these novel chemicals can be commercially and effectively used for delaying the ripening of perishables, possibly beyond the 14 days, with retention of the optimum quality character, 12 to 14 days, more than that one. Further studies with respect to the application of these novel chemicals at various concentrations combined with storage, low and ambient temperature in order to delay the ripening of various fruits and vegetables needs to be standardized for their commercial exploration. Therefore, each fruits and vegetables is sensitive to the 1MC for specific chemicals. Therefore, we have to standardize or we have to optimize the dose. Then we have to decide the exposure time. Then we have to storage temperature. These are three crucial points. We have to optimize that one. Then definitely each fruit, perishable fruits or horticulture produce can have the longer shelf life. These are the references. Thank you very much for your patient listening. I
Yes,
tried
sir.
my best to cover most of the points.
Yes, sir. Yes.
Thank you very much, sir. It was really very nice presentation and very knowledgeable to all the participants too.
Yes, sir. Yes. On behalf of Department of Technology,
So you can just have some questions
hearty
to.
thanks to the Dr. BK Sakhale, sir. Professor Dr. BK Sakhale, sir. Now we are going to begin the answering the questions submitted during today's presentation. As a reminder, you can still submit the questions through the chat box. There is one question from the participant, Dr. Mohan Kumar sir, is up to what concentration the AgNO3 or AgNP is or can be used for the treatment?
Sir, it depends upon the particular type of fruits or vegetables. In
Yes.
case our particularly in tomato, 1 micromolar we have selected, and that will found the effective as compared to as followed by the 1 MCP. So it depends upon the type of fruits and vegetables. So that's why I have explained that trial and error experiment, we have to do a lot of experiment it requires. So we can decide on the basis of the particular fruit and vegetables, on the basis of their physiological nature or morphological characteristics, how it respires. So it is whether it is climateric or non-climateric. So in climatic fruit, the dose will be higher. In non-climatic fruit, the dose will be lower.
Yes. Yes. The second question from the same sir, Mr. Dr. Mohan Kumar sir. How to remove the ethylene and antagonists after the post-harvest processing?
The ethylene can be removed by using a number of the ways. So the ethylene antagonist function, that one thing we are having the capacity to regulate the ethylene or to stop the synthesis of the ethylene, or if ethylene is produced, there are number of the ways to absorb it. So the in smart packaging also, ethylene can be absorbed. In potassium permanent tablets are also kept that you can also absorb the ethylene. So number of the ways are there. Even availability of ethylene, they will not do anything if you apply the 1 MCP, because the whatever the sites are, binding sites are, that will be bound by the 1 MCP. Therefore, ethylene will have nothing to do with these fruits because they don't have their sites to act.
Yes, the same question. The next question is, does that ethylene require some special treatment to remove? Means same. Yes, I think you have answered the
Yes, 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 same,
question.
same
Yes,
answer.
yes,
Yeah.
same as answer. Are there any questions from the participants? You can unmute yourself and you can ask the same and participate in this today's FDP. I think you have covered all the details, means all the details correctly, so that there are a few questions from the audience. You have covered all the details systematically.
Thank you, sir. Thank you.
Yes, yes. Now, Dr. I.S. Uchan sir, if you want to add something, you can add. Or if you want to ask any questions, you can ask.
Sir has given much information about all
Yes.
the processes. It was really, that means I saw the presentation. So much
Yes.
information was there. That means much knowledge we have already taken from that one. And even I think regarding various this one more question was there. Up to what concentration this AgNO3 can be used in the treatment? That was one more question that is posted by another
Yeah, I
participant.
already answered the
Answer
question.
same
So it
question.
depends,
Yes,
yeah.
answer is the same.
Yeah. Yes, sir.
You
now,
can send me the email if you have any query. You can write to me.
Yes, sir.
yes,
My detailed contacts are available with this organizer.
yes,
I would
Yes.
like to express my sincere thanks to the Irana sir for giving me this opportunity.
Yeah, actually, I myself thank you very much because very first call was to you and you accepted, and from that one this whole process started. So actually, I am thankful, and you are with us from for many works with us, and Atal is one of that one. So I am very much thankful from our Department of Technology and Surveillance to Kolhapur for accepting and delivering such a nice lecture to us.
Thank you, sir. Thank you.
Thank you, sir.
yes, yes. On behalf of Department of Technology, Shivaji University, Kolhapur, and all the participants, I would like to give a special thanks to Professor Dr. BK Sakhale sir. And thank you everyone for attending the fifth session of online faculty development program entitled Recent Trends in Food Processing Preservation Technologies. If you have any other questions, please contact to our coordinator, Dr. I.S. Urchan sir, Ms. Coordinator of this FTP. We will meet, Iran sir, if you want to announce anything, you can.
No, we'll meet for the next lecture by Dr. Prabodh Haldi at 3 o'clock. So after
Yes.
that one, we will have the lunch break.
Yes.
Thank you once again, Dr. BK, Professor BK Sakhale sir, and all the participants. Okay, so we'll meet for the next lecture at 3 o'clock.
At 3 o'clock. Means
Yeah.
this is a lunch break right now.
Okay, so this will be lunch.
Have a good day.
Okay. So those who want to give that regarding the feedback and all this, now it is already posted. Okay, so you can give that, fill the link. Okay, one more time I will just post it again and then we will close the session.